Okay, we're going to be picking up at vaccines in chapter 21. The question in the notes is explain what a vaccine is and how they work. So this is what the book says they are. Some vaccines are dead viruses or bacteria. Some of them are weakened, which is attenuated. That means that they are still alive, but they won't make the average person sick. The newer vaccines for COVID are what are known as mRNA vaccines, and those work a little bit differently. Sometimes vaccines are toxoid vaccines, which means they are providing the molecules that are going to help you against toxins as opposed to viruses or bacteria. So there's different vaccines for different reasons, essentially. For the how they work. What vaccines are supposed to do is help you mount an immune response against something that was going to harm you. They do that by helping you make your own army of B cells that are going to differentiate into plasma cells to make antibodies and memory cells that are going to be long-lived. This only works assuming that the antigen is both immunogenic and reactive. The benefit of a vaccine is that while you are mounting your own army, you won't develop any symptoms of illness usually. Now, there can be some side effects, pain in the arm, you can develop fevers in some cases, but I'm going to bet that the majority of you have received the polio vaccine and that the vast majority of you do not have polio, and that's because the vaccines prevent you from getting the symptoms of the actual illness that they are protecting you from. Vaccines are one of the things that I tend to get very soapboxy about. They are a very, very important tool in terms of managing public health, and there is a lot of misinformation out there when it comes to vaccines. So that's why I tend to get soapboxy about it. So again, what they do is allow for the antigen challenge because there is at least an antigenic determinant within the vaccine. So we're still going to do the come at me bro sort of a situation, assuming that you have immunogenic cells that are in there and capable of binding to whatever the epitope happens to be. We're going to accept that antigen challenge and we're going to do the clonal selection thing and again make your army of B cells. We haven't talked about cellular immunity yet, and vaccines don't really ramp up cellular immunity. They really just do humoral immunity. So that means they're really only activating half of your specific immune system. So they're really good at providing you with an antibody response, but not really good at providing you with a cytotoxic T cell response. Now, because mRNA vaccines are new, I did want to stop and explain those just a little bit. The books don't even talk about those yet because, again, they're brand spanking new. So here's why I have to throw you back to AMP1 to remind you about the central dogma of biology. This was protein synthesis. We talked about this in chapter 3. Remember, your DNA tells you how to make everything. Your RNA is copies of genes, and mRNA is specifically messenger RNA. It's a copy of a specific gene or some specific genes that are useful in the same pathway. You make RNA from DNA during the process of transcription. Then the RNA goes out and it finds the ribosome. The ribosome will read the RNA and then will make a protein reading the recipe from that RNA. So when you get an mRNA vaccine like the Pfizer or the Moderna vaccine, you're getting a shot of mRNA. So what's going to happen when you get that shot is your body is going to read that mRNA and it's going to make the protein that mRNA codes for. What that mRNA codes for is the spike protein. So this is what coronavirus looks for, SARS-CoV-2. All these little red things on the surface are the spike protein. So it's a protein. This is the part that is immunogenic and reactive in our body. That little spike protein has a receptor binding domain that is going to bind to the ACE2 receptor in our body. That's what allows it to get into your cells and then cause all the rampant problems that it's going to cause. So the mRNA codes for that spike protein, which allows you to again accept the antigen challenge. The benefit of this vaccine is mRNA does not last very long in your body. You get rid of it all the time. So you're not going to keep making spike protein. And here's the other advantage. You only made spike protein. You didn't make the whole SARS-CoV-2. You just made spike protein. That means you never had virus replicating inside of your body. And so the risk of side effects was incredibly low. And in fact, with the mRNA vaccines, we saw very few side effects with that, just really pain on the arm. The symptoms that came along with that, like the fever and whatnot, that tended to go with your immune response ramping up because of the spike protein that you were making, not because you had a viral response or anything like that. Now, the Johnson & Johnson vaccine, that was a whole different type of vaccine, and that's why it had different side effects from what the mRNA vaccines were going to have. So what I want to make sure you guys understand is, while I, again, will get very soapboxy about vaccines, pardon me, and tell you how very helpful they are, I'm not going to tell you there's not any risk associated with those vaccines. All interactions have some sort of risk associated with them, but the mRNA vaccines are very safe and they are very effective. And so I just wanted you to understand how those work and why they are so safe. And it's because the mRNA doesn't stick around in your body. It just stuck around long enough that you would make the spike protein and then make your own memory cells. And the memory cells are what are supposed to stick around for longer. Unfortunately, we're seeing that you're not really making a very strong memory army. And so that's why we're seeing that you need boosters over time, which is why booster shot after 6 months of the first 2 doses is what's currently being recommended. Okay, now getting back on my soapbox, vaccines absolutely work. So getting my snarky little, have you seen smallpox lately? No, nobody has, because it was eradicated back in the 1970s. There have been no wild cases of it since the 1970s. There are countries that still have this in their freezer, which is a little bit terrifying, especially because one of those countries is currently at war with Ukraine. This is weird that I record this because at some point you might watch this in the future and hopefully this war will be over. God, it better be. But it's not something you see because vaccines eradicated that disease. We are so close to eradicating polio. It is only endemic to 2 countries right now, and it happens to be 2 war-torn countries, Pakistan and Afghanistan. There's also a vaccine-derived outbreak in some countries in Africa, and here's what I mean by that, and here's what I'm going to come back to a couple slides ago. So notice that I said that vaccines can have full-on dead pathogens or attenuated pathogens. Well, there's a couple of different versions of the polio vaccine. If you get the polio shot, that has dead polio virus in it. You cannot get polio from a dead polio virus. There are also oral polio vaccines, which is somebody squirts a liquid into your mouth, and that one is an attenuated polio virus. Now, the average person is not going to get polio from that, but if you give it to a person who has a weakened immune system, they can actually develop polio from that. Now, we intentionally give oral polio vaccine to certain countries in Africa because what it will do is actually reproduce within the people that we give it to, and then it will leave the body as it normally does, which is in the feces, and then it will get into the water and then help sort of drive immunity within the population with the attenuated, very weak polio. So vaccinate one person and you're kind of vaccinating all the people that drink water downstream of that person. And I know that's a horribly gross thing to think about, but it's the way that it works in those very rural areas in Africa. The problem is, because it was a weakened polio but still alive, it mutated and went back to a more virulent strain that caused full-on outbreaks of polio in some places. And so again, viruses absolutely do have risks, and that's something that you do have to be aware of. But again, here in the United States, we use the dead polio shot, and so there is not a chance that you are going to get polio from that. I want to make sure you are 100% clear on that. This is what the oral polio vaccine looks like, and again, just me telling you, yeah, there's risks. So there are certain vaccines that you have to get when you're going into school, and a lot of those things are illnesses that you will probably never see in your life. Like, probably you guys have never met a person with polio, because again, it's been eradicated in the United States. There's only a few countries that still have it. You've probably never seen a person with tetanus, because it's incredibly rare to see a person with that, because we vaccinate against tetanus. It's part of the Tdap or DTaP vaccine. As more people stop vaccinating because of the vaccine hesitancy or full-on anti-vax movement, we're starting to see diseases reemerge that we had nearly eradicated, like whooping cough or measles. In this case, this is a child who has tetanus, and tetanus is one you do not want to mess with. It will make your muscles contract so hard, it will break the bones in your body. It's just not one that you want to play around with. And so personally, I would take the risks of a vaccine over the risk of getting tetanus every day of the week. But that is, again, something that every patient has the right to decide what risks they're willing to take and which risks they're not. It is your job, if you're going to be working in the medical community, to give them the proper education they need so that they can weigh those risks correctly. When my son was getting his vaccines ages ago, we actually had the choice between, do you want your kid to get the polio shot or the oral polio vaccine? And to me, that was an easier choice because I didn't want to give my kid a shot. You know, little kids hate shots. So we went with the oral because he was a healthy kid. The chance of him getting polio was very low. However, if he had an autoimmune condition or he were immunocompromised, would have absolutely done the shot. And so there's calculus involved in it, essentially, but you got to do informed consent, essentially. Okay. Hopefully that helps you see a little bit of what goes on in a person's head as they're trying to figure out what they want to do. Now, going back to finishing up humoral immunity at this point, you have compare and contrast active humoral immunity with passive humoral immunity. So active. Here's what I really need you guys to try to link this to is what I'm saying on this slide. So I'm going to highlight in a nice little yellow. Remember, this is all about the army that is helping you fight. And if in active immunity, you build your own army. You have the genes to make it, you made it, which means you also made your memory cells. In passive immunity, on the other hand, you do not make your own army, which means you did not make your own memory army. Instead, you borrowed somebody else's army. So you didn't die, you got better, but you are not immune in the case of passive immunity. And so I need to make sure you understand that. So active immunity, you made your own army. Passive, you borrowed somebody else's. Okay, I gave you this picture to help you differentiate between the different types. Wonder why I have that sticky note there. Don't you love when you find sticky notes in your... Wonder what I was trying to remember. Okay, sorry. So in either case, active or passive, you can naturally acquire active immunity or you can artificially acquire active immunity. Naturally active immunity is you got sick and you got better. When you got sick, you learned how to make your own army. Your army protected you, but you got symptoms, you were unwell, and then you got better. Artificial active immunity is we gave you a vaccine to teach you how to make your own army, but because it was a vaccine with a weakened or dead or mRNA thing, you learned how to make an army without actually developing symptoms because the thing couldn't reproduce inside of you at the time. So you learned how to make the army without getting the symptoms. Now, passive immunity is, again, you borrow somebody else's army, and there's two ways you can do that. Naturally, borrowing an army is basically you get it from mom. So you get some that cross the placenta while you're still in utero. And then if you breastfeed, you get some from mom that way. Or if you're the breastfeeder, you give some to your baby that way. Artificially acquired is you get an injection. So if you're getting serum, like they talk about convalescent plasma, you're getting plasma from a person who was sick, so they've made their own antibodies. That's going to help you get better, but that's not going to help you make your own army. You were just borrowing somebody else's army, somebody else's snipers, somebody else's bullets to fight the infection. So that's how that works. So we can give you, like I said, convalescent plasma. We can give you concentrated antibodies. They're usually called monoclonal antibodies. There's different ways that we can do them. So is this calf receiving active or passive? Well, since we're borrowing from mom, that would be passive. Okay, this one is a big one. There are, again, not only can mom pass her antibodies to the baby, mom can also pass some viral infections over to the baby. Hepatitis B is one that can actually cross the placenta. So one of the things that we try to do is protect the baby while the baby is still inside mom to make sure the baby does not come out infected with hepatitis B. So we can give the baby immunoglobulins while still inside mom so that the baby can borrow an army to not get Hep B while still in mom. So what's the question I'm asking? Will the baby be immune to hepatitis B once born? No, because the baby was just borrowing an army. We're still going to have to vaccinate that baby against Hep B once we are ready for that after birth. Okay, now this one is one I always like to tell because we live in Texas. Here's the basic backstory. You hear some really weird things in relation to snakes. One of the things that I have been stressing is in order for you to be immune to something, the something has to be immunogenic. In order for something to be immunogenic, you have to have a gene that allows you to make an army against whatever that something happens to be. And there are not humans that are immunogenic to snake venoms that are out there, at least not the vast majority. There's probably some mutant human out there that does. But, okay, so this little guy, I was visiting my parents' house and saw this little guy on their bird bath, probably waiting for some bird to come by. And hopefully you guys are all sitting there going, oh my God, it's a copperhead, because I want you to know that is a copperhead. It is a venomous snake. Humans cannot make antibodies against copperhead venom. So if you get bitten by a copperhead, you should go to the emergency room. Here's the story behind this kid. This kid had been sent out to go coil a hose, accidentally grabbed the snake, not this snake, but you get my point, hopefully, accidentally grabbed the snake and tried to coil a snake with the hose, and the snake did not like that very much, and so the snake bit him, and you can see we've already got a reaction that is starting right here. So snakes can sometimes do dry bites. If they do that, we got to look out for infection, but you're probably going to be okay. But here, this is not a dry bite. We've already got a reaction that's starting. So this kid is going to get antivenom. Now, here's how we get antivenom. Antivenom is actually antibodies that were produced by a different organism. The way that we collect that is we milk snakes, which is a fancy way of saying we collect the venom from snakes, and then we take that venom and we inject it into horses. The reason that we do that is horses are genetically capable of making antibodies against snake venom. So it won't hurt the horse. They are immune to snake venom once they've been exposed that first time. So once they make the antibodies, we can take the horse blood, separate out the antibodies, and that's what antivenom is. It's the horse antibodies. Then we inject that into the person as antivenom, and then after lots and lots and lots and lots of doses, more than likely, hopefully we will be able to save your life. So that is a case of artificially acquired passive immunity. We borrowed an army from a horse in this case. So if this kid gets bitten again by another copperhead in the future, he is not immune to that snake. In fact, he is more likely to get more sick the second time, the third time, the fourth time, because as it turns out, the more often you get exposed to something that is reactive but not immunogenic, the more sensitized you get to it. So here's the story behind this guy. I always forget his name. I wish that I could remember it, but he is one of those what do you call them, like evangelical preacher people that would put up a tent on the outskirts of town. And he used rattlesnakes in his, God, again, what would you call it? Not sermons. This is so sad. I'm not a person who goes to those things, so I don't know, but whatever. In his shows, would you call it a show? He would allow the snakes to bite him because he believed that God would cure him. Well, the first several times, he was okay, but each time he got bitten by a snake, he got sicker and sicker. I think fourth time he got bitten, he passed out. The congregation tried to take him to the ER, but he came to before the ambulance could take him away, and he ended up declining medical service, and he went home and convalesced for a while. And then, stupidly, he went and did it again. The fifth time killed him. He is dead now because the more you get exposed, the worse it is for you. There are people all over the state of Texas who believe you can become immune to snake venom. In fact, if you go to a place in West Texas called Sweetwater, I think it's spring, they do the Sweetwater Rattlesnake Roundup, and you will find people there who will drink rattlesnake venom because they believe it will make them immune to it. And no, it won't. All it's doing is making it more likely that they die when they actually get exposed or bitten by a rattlesnake. And that is because you do not have the genetic capability of becoming immune to rattlesnake venom. So that's the reason I wanted you guys to get that story. Your immune system is based on your genes, and if your genes don't allow you to make the army or make the antibodies, you're never going to be immune to it. And so that's an important thing for you to kind of understand there. All right, I'm going to skip past these questions. Next up, antibodies. I'm talking about them a lot. They are also called immunoglobulins. That is a word I threw out there earlier. So globulin is just a fancy way to say wonky shaped protein, essentially, and immuno because they're functioning in the immune system. Antibodies are always based off these little monomers that are Y-shaped, so Y-shaped proteins. There are 5 classes, and your book gives you the mnemonic MAG to help you remember them, because there's IgM, IgA, IgD, IgG, and IgE. The Ig always stands for immunoglobulin. Okay, so here's the basics. IgM, it's a pentamer. What that means is it's actually 5 Y-shaped proteins all put together. What that means is, so the tip of each Y, I'm going to change my color since I'm doing this on a white background at this point. This is where we can bind to one antigen. This is where we can bind to another antigen. So this IgD can only bind to 2 antigens at one time. This IgM, on the other hand, it can bind one here and one here and one here. So it can actually bind up to 10 antigens at one time. It is very efficient at picking up a whole bunch of antigens at one time. So this is the first antibody class released once your body realizes there's a problem. It's the cleanup crew, essentially. Okay, IgA is a dimer. It gets released into your mucous membranes. There's actually a bunch of research going into trying to create a nasal vaccine against COVID that will help trigger IgA vaccine responses, because it's believed that that will help actually trigger better immunity. There will be nasal vaccines as opposed to shots. They're working on that. I'm looking forward to that because that would actually be better for us overall. IgD is a monomer, and what's special about that is it's actually small enough. Well, I'll get to that later. It's on the surface of a B cell, and this is the receptor for the antigen challenge, is the way that I want to say that. So when we do the whole come at me bro thing, this is the part that comes at the antigen. IgG, it's also a monomer, and it's small enough that it can actually cross the placenta. And so when we talked about the naturally acquired passive immunity that you can get from your mom when you're still inside of her, this is the antibody that gives that. And then IgE is usually one that you only secrete in very small amounts. However, in industrialized nations, turns out we upregulate these and produce a bunch of them, and they trigger allergy responses. Your book says that antibodies have a plan of action. What does it mean and how do they work? Here's what the plan means. The P is for precipitation, the L is for lysis, the A is agglutination, and the N is neutralization. And here's what we mean by all of those. I like this picture. It's showing you what's up. So starting with the P for precipitation, it means that we are going to clump molecules together. If a bacteria or a bacterium is the singular for bacteria, or a toxin produced by a bacterium is floating around, we need to clump that toxin. That's what precipitation does. So this is really good at clumping toxins together, is what I want you to know about precipitation. Better for toxins. This would be good against venom if we could make antibodies against venom, but we can't. When we take antivenom, this is how antivenom works. L was lysis was next. Sorry, I'm doing this in the same order as plan. So cell lysis, the antibodies can't do this on their own, but with the help of the complement system, that's these proteins up here, antibodies are going to help the complement system poke holes in the cell. And holy cell is a lytic cell, so it's going to pop the cell open. That's how antibodies help. It is important that you understand they can't do that by themselves, though. So they pop the cell with the help of the complement system. A for agglutination. So y'all know what agglutination is. It's very similar to precipitation, only we are clumping cells together, not just toxins together. So this would be us clumping bacteria together or protists together. Neutralization. So remember I was talking about that spike protein earlier? Spike can't get into your cell if spike is covered up. And so if the antibody covers up the spike protein, spike can't interact with ACE, and now your problem is solved. And so that's how neutralization is going to work. That is where I'm going to stop this video.